Welcome to the first video of Attempting Science YouTube series. In this first video, we will explain the meaning of the broad concept synthetic biology in a clear and comprehensive manner. Synthetic biology is the field where biology is combined with engineering. Have you heard of GMO, for example? Then you know it is an organism where the DNA has been altered. This is made possible by synthetic biology. The field also covers the pharmacological harvesting, where, for example, synthetic insulin required in diabetics can be manufactured by letting bacteria produce the protein. In the wild, bacteria cannot produce the human form of insulin. But scientists have succeeded in isolating the genetic code for insulin in humans. They have then altered the DNA to a form that is readable by bacteria, and lastly transformed into them. We have found something that's not inherently produced by bacteria and made them produce it anyways. Just like humans, bacteria have a long strand of DNA. However, most bacteria do have cyclic DNA compared to our biological code that is linear. Bacteria also have plasmids, which are small circular DNA parts with information that easily can be exchanged between adjacent bacteria. The plasmids can be extracted and modified so that we can insert a gene from another species in order to, for example, produce insulin. One way for scientists to do this is to let enzymes work as scissors, and the plasmid can get cut open by a double-stranded break in the DNA. The gene of interest can then be placed inside the broken plasmid, which is then sealed by another enzyme called ligase. The bacterial culture will then be placed in 37 degrees for a number of hours depending on which species of bacteria that were used. After the bacteria have grown colonies, you can add inducers to put the protein production on. However, many forms of synthetic biology has been questioned when it comes to ethics. It requires a lot of responsibilities when one is meddling with nature. As of today, creation of life from nothing is still improbable to happen, but we can do a lot of other things. You might have heard concerns about so-called designer babies. If you have not, it is the idea that one can take a fertilized egg and edit its genome. In this way, the parents could decide which genes to turn off and which genes to turn on. We have done this to bacteria and animals for a long time, but doing it to humans is a lot more controlled and controversial. However, in March of 2015, a scientist took it into his own hands to, in secrecy, edit the first human embryo in order to create a child that would inherit a gene that makes it unable to get infected from HIV. Normally, most people carry a gene that encodes for a specific receptor on the white blood cells that the HIV virus targets to get inside and duplicate. Even though the gene makes us susceptible to the HIV, it is probably not there for that particular reason. In fact, many genes perform their functions together with numerous other genes, and you cannot know which effect it will have to turn off a gene or add another. Since then, harder regulations have been discussed, but also the future prospects of well-controlled further research. There are three major concerns of the future rise of human, animal, and genome editing. Firstly, there is an opinion that creating a new life form or life is bad or leads to bad consequences. Secondly, the regulations of the future will either be led by unwarranted fear or unfounded optimism. And lastly, the new technologies need public approval to be able to proceed smoothly. So where will the field of synthetic biology be taken from here? At least when it comes to editing of bacteria, the science will probably proceed as it is today. And when it comes to crops, animals, and even humans, Only the future will tell us what happens there.